Hello students, today we are going to see the second part of the lesson, classical genetics. In the first half we have learned about Mendelian genetics, monohybrid, dihybrid, trihybrid crosses, reciprocal backcross and test crosses. Now let us roll over into the intro and get into the class. Now let us recall the Mendelian laws of inheritance. The first law is law of dominance. The second law is law of segregation or law of purity of gametes. The third law is law of independent assortment. Law of dominance. What is law of dominance? When two pure breeding varieties with contrasting traits like purple flower and white flower were crossed, only one of the trait will be expressed in the offspring. The trait which is expressed is referred as dominant. So purple color is expressed in the offspring, and so this expression is referred as dominant, and which is not expressed in the F1 generation is referred as recessive. Law of segregation. In an organism at the time of gamete formation, the allelic pairs of the genes segregate. As a result, each gamete will have only one allele. For example, the allelic pair T, they segregate at the time of gamete formation, and so each gamete will get one of the allele. Similarly here, each gamete got capital T, one gamete got capital T and another gamete got small t. So this is the principle of segregation. So chromosomes replicate capital R, small r, small r. The gametes are formed with the alleles, each allele getting into one of the gametes, and so four gametes are produced at the end of the meiosis II. Law of independent assortment. This is the third law. So during gamete formation, the allelic pair of one gene will sort out independently of the alleles of the other gene. For example, if the individual is going to have we are going to consider two types of genes, that is R gene and Y gene. The allelic pair capital R, small r, and the Y gene allelic pair capital Y and small y. The allelic pair of one gene will sort out independently of the alleles of the other gene. So the inheritance of R gene is totally independent of the Y gene, as well as the inheritance of the Y gene is totally independent of the R gene, and the allelic pair of each gene also will sort out independently, and so the individual is able to produce four types of gametes. Now let us learn gene interaction. What is gene interaction? Interaction of the alleles of the genes to control a phenotype. Phenotype is a character. So interaction of the alleles of a particular gene to control a phenotype is said to be gene interaction. And this gene interaction was first studied and explained by W. Bateson, and he has classified this into two types of gene interaction. That is, first type is called as allelic gene interaction or intragenic gene interaction. The second one is non-allelic gene interaction or intergenic gene interaction. Now let us see one by one. First one is intragenic gene interaction. This is the interaction between the alleles of a single gene, and it is said to be intragenic interaction or allelic interaction. So only one gene is considered, that is color of the flower, red color, capital R gives the red color, whereas when the recessive form of it, small r gives the white color flower, whereas the heterozygotic condition, capital R and small r, give the intermediate color, pink color between the red and the white. So this is what is intragenic gene interaction. Because of the alleles, different types of alleles of the particular gene, the expression of the phenotype also differs. Now what is intergenic gene interaction? Intergenic gene interaction is studied in the fruit called summer squash fruit. So the interaction between the two genes like W and G gene. So W gene represent the shape of the fruit, whereas the G gene will represent the color of the fruit. So when the capital W or the dominant alleles of the W are present, then the fruit would be round in shape, and along with the capital G, that color gene, it is white in color. So both the gene in dominant alleles, they produce a round shaped white fruit, whereas shape gene, that is capital W becomes the small w when both are small w with the color of the fruit, that is white color, capital G dominant color, so it becomes yellow in color. So because of the presence of the W, small w gene, the color is expressed as yellow. So the presence of the small W gene alleles makes the color of the fruit as yellow in color. So this is what is called interaction. Presence of one gene affects the expression of another gene. This is what is interaction or intergenic gene interaction. So similarly, when both the alleles become the recessive type, small w and small g, then the fruit produced would be green in color and it will be of thin shape. The shape will not be round. So this interaction between the alleles of different gene is said to be interallelic gene interaction or non-allelic interaction. So next we shall learn the incomplete dominance. What do you mean by incomplete dominance? The intragenic interaction can be of 4 types, and one among them is incomplete dominance. The 4 types of intragenic interactions are the incomplete dominance, codominance, multiple alleles and pleiotropic genes. So in that first one is incomplete dominance. That means there is no blending of any character or genes, and this was first studied by the German botanist Karl Correns in 1905 in his experiment with the 4 o'clock plant or Mirabilis jalapa. So he has taken 2 pure breeding variety plant, that is a red flower which is having the gene R1, capital R1, whereas the white flower producing plant, that is R2. So when he crossed both in the first generation, in F1 hybrids where seems to be pink flowers, that means it is in the heterozygotic condition. So the R1 and R2 combine to form the pink color, which is becoming the intermediates. So when this red flower and white flower are crossed in F1 generation, it produces an intermediate phenotype pink color flowers. When pink color flowers of the F1 generation were interbreed, when they were selfed, then in F2 generation they were able to produce all the 3 types of flowers, that is a red color flower as well as the pink flowers and white flowers. Here there is a law of segregation applies. The phenotype and the genotype ratio will be the same, but there is no blending of the genes. In F2 generation, R1 and R2 segregate and recombine to produce red, pink and white color. Here there is no blending of the genes. In the F2 generation, the R1 and R2 genes will segregate and recombine to produce the red, pink and white in the ratio of 1:2:1. The R1 allele will code for an enzyme that is responsible for production of the pigment, whereas the R2 allele will not code for any enzyme and it can produce only defective enzyme. And so R1 and R2 genotype produce only enough red pigments to make the pink color flowers. So the 2 R1 R1 is needed. 2 R1 R1 alleles are needed for producing red color, whereas 2 R2 R2 genes are needed, alleles are needed for white flowers. So if the blending had taken place, the original pure trait would have been lost and it would not reappear in the F2 generation. So there was no blending, and so the parental colors are produced in the F2 generation. It is very clear that Mendel's particulate inheritance take place in this cross, and this is confirmed by the reappearance of the red color and the white color flowers in the F2 generation. Now let us learn codominance. Codominance is a type of inheritance in which both the alleles of the same gene expresses their phenotype in the heterozygous condition. For example, R1 and R2, both the alleles in the heterozygous condition will be expressed in the flower. So the phenotype of both the alleles are expressed equally without any blending. So neither of the allele is considered as recessive because both are expressed in the F1 generation. So for that example, codominance in Camellia species, the red flower and the white flower when they are crossed. So both the alleles are expressed in the heterozygous condition equally in the F1 generation. When F2 generations are produced by selfing, so the characters, three characters are expressed like intermediate as well as the parental characters are expressed in the ratio of 1:2:1. So phenotypic as well as the genotypic ratio becomes the same 1:2:1. So this is a comparison between codominance and incomplete dominance. In the codominance, so red and white color both are expressed in the F1 generation without blending, whereas in the incomplete dominance, so the intermediate is produced between the red and the white, the intermediate is produced as the pink color. The other examples of codominance can be sickle cell hemoglobin and ABO blood grouping system in human beings. So in human beings, IA and IB alleles of the I gene both are codominant, and which also follows the Mendel's law of segregation. Now codominance can be demonstrated in the plants like Gossypium. The codominance in the seed proteins of Gossypium species. The electrosporosis of the seed protein of F1 hybrid between Gossypium hirsutum and Gossypium strutianum gives an additive band pattern indicating the codominance. The heterozygotic genotype give rise to a phenotype which is distinctly different from either of the homozygous genotype parents. So F1 heterozygote produce F2 progeny in a phenotypic and genotypic ratio of 1:2:1. Now what are lethal alleles? An allele that has the potential to cause the death of an organism is called as lethal allele. And most of these lethal alleles or genes are recessive. Edwin Bohr was a German scientist. He explained about the lethal genes in Antirrhinum plant. So Antirrhinum plants, this is a picture of it, and they are commonly called as snapdragon plant. Now let us see the different types of Antirrhinum plants. Types of plant with green leafed plants and golden leafed plants and white plants. So pigment present in the green leafed plants chlorophyll, and so the genotype would be capital C, capital C. And the pigment present in the golden leafed plant is carotenoid, and the genotype would be capital C, small c. And the white plants will not be having any pigment or no chlorophyll is present, and the genotype would be small c and small c. So recessive lethality is observed in the snapdragon plants. So Antirrhinum aurea is the plant which is having the genes in the heterozygous condition, that is capital C and small c. So when two Antirrhinum aurea plants are crossed, so they are able to produce white flower, white, yellow and green plants, and the ratio of 1:2:1. This is due to the lack of the chlorophyll. White plants fail to survive. They will not be able to survive because they cannot produce the starch and they cannot survive. And so the ratio will be modified as 2:1. Now pleiotropism. A phenomenon where one gene influences more than one phenotype in an organism is pleiotropism. And Mendel observed pleiotropism in pea plant. He noticed purple colored flower, brown seed and dark spot in the leaf axil. All these three characteristic features are present in the same plant, and they are contributed by a single gene. Similarly, when he crossed this plant with white flower, light colored seeds and no spot on the axis of the leaves, so all the other three traits were also seems to be inherited together as a single unit. And this is due to the pattern of inheritance where the three traits were controlled by a single gene with dominant or recessive alleles. So this is the example for pleiotropism. And another example you can say in animal sickle cell anemia. Now intergenic gene interaction, dominant epistasis. In dominant epistasis, An allelic pair of a gene present in one locus, one point of the chromosome, will mask the phenotypic effect of another allelic pair of a gene present in another locus. So you know how the genes are present on the chromosome. So an allelic pair of genes present in one locus of the chromosome will mask the effect of phenotypic effect of another allelic pair of gene present in another locus, and this is what is called as intergenic gene interaction and dominant epistasis. The gene which masks the phenotypic effect of another gene is called as epistatic gene. The gene that masks the phenotypic effect of another gene is called as epistatic gene, and the gene that is masked from expressing the phenotype is called as hypostatic gene. Now let us see the dominant epistasis in summer squash fruit. Two genes are present in two locus where the presence of the alleles of one gene will mask the effect of the other phenotypic expression. So gene W, capital W, is dominant. When this one of the allele of this gene is present itself, the fruit will be white in color. The next gene G, capital G, if it is present, the fruit would be yellow in color. But this white is dominant over this G. When one of the allele is present itself, even if both the G alleles are present, capital G are present, the fruit will be white in color. That means white is dominant over yellow color, and a similar way, and yellow will be dominant over green color. So in a cross between the white fruit and the green fruit, the white fruit will have two WW, capital W genes, whereas the green fruit should have small ww and small gg only can become the green fruit. Or you can even cross a yellow fruit which is having small w, capital G and capital G can be. So when you are crossing these two pure breeding variety, you will get in F1 generation heterozygotic individual, that is capital W, small w, capital G and small g, which would be also white in color because one allele is enough, one W capital dominant allele is enough for producing the white color fruit. When this is selfed in F2 generation, the gametes produced can be written here, and you will be able to get the phenotypic ratio, that is white fruit, yellow fruit and green fruit in the ratio of 12:3:1. So here when capital W, one of the gene is present also, the fruit will be white in color even though it does not mind the two capital G. So white is dominant over yellow. In the similar way, if two G capital G's are present alone, capital G, one gene is itself, one allele itself is enough to produce a yellow color. And only if all are recessive gene only, it will produce a green color fruit. Next we have polygenic inheritance. Polygenic inheritance refers to the inheritance of single trait controlled by more than one genes. That means one characteristic feature will be controlled by more than one genes, 2-3 genes. In simple term, many genes will contribute together for determining a single character. So like this, G, W, E, Q, all these genes will be contributing for a single character. Herman Nilsson Helle, he was a Sweden genetist. In 1909, he demonstrated the polygenic inheritance in wheat kernels. Polygenic inheritance in wheat kernel. Wheat kernel color is controlled by two genes, namely capital R1 and capital R2. And this capital R1 gene has two alleles. That is, capital R1 is dominant form, whereas the small r1 is the recessive form, which is white in color. So R1 will have two alleles. R2 has two alleles, R2, capital R2 dominant form, whereas small r2 will be recessive form, which is white. So the dominant form of the gene R1, R2 gives the red color, whereas the recessive form of the gene small r1, small r2 gives a white color. So here in a cross between the two parent, that is capital R1, R1, capital R1, capital R2, capital R2, when it is crossed with small r1, small r1, small r2, small r2 white kernel rice, then in the F1 generation the plant produced will have medium red color kernel. And when this F1 is selfed for producing the F2 generation, we will get different varieties of kernel color, and that is based on the number of capital R allele present in the particular individual. So this is the color of the kernel is determined by the number of capital R genes. So if the four R genes, capital R genes are present, like in the first case, the kernel color would be dark red. Whereas in 3 capital R's are present, then the kernel color would be medium dark. And 2 capital R are present, then the color of the kernel would be red. And if only 1 capital R is present, it would be light red. And all are recessive alleles, like all will be small r, means it will be white. So, since the phenotype is determined by the number or the count of the genes, it is called as it is an example for a kind of quantitative inheritance. And so the phenotypic ratio produced also would be 1:2 dark red, 4 medium dark, then 6 would be red color, and another 4 would be light red, and last one would be white color kernel. Now let us learn about the extra chromosomal inheritance. Chromosomes are present in the nucleus, and these chromosomes will possess the DNA or the genes. There are also DNA and genes which are present in other cytoplasmic organelles such as mitochondria and chloroplast in the plant cell. So when the genes which are present in these organelles, when they are expressing a phenotype in the plant and when they are inherited to the offspring, we call this inheritance of characters or called as extranuclear inheritance or cytoplasmic inheritance. The inheritance of characters that are controlled by the genes which are located in the DNA other than chromosomes of nucleus. It is called as extranuclear inheritance or cytoplasmic inheritance. In this type of inheritance, the traits are controlled by the genes of the DNA of the cytoplasmic organelles like mitochondria and chloroplast. Chloroplast inheritance of leaf variegation in Mirabilis jalapa. So here in this cross 1 and cross 2, first cross, we are taking a male plant with pale green leaf and the female plant with dark green leaf. When they both are crossed, the offspring in the F1 generation will be dark green leafed plant. Why? Because the genes which are responsible for the color of the leaf is present in the chloroplast, and that is contributed by the female plant alone. During fertilization, the male gamete from the male gamete, the nucleus alone will be entering into the ovum to fertilize the ovum, the egg cell. And so the contribution of the chloroplast from the male is absent here. And so the chloroplast that is containing the gene for the leaf color is contributed only by the female plant, and so the individual, the offspring which is produced out of these two will be only from the female plant, that is dark green leaf. The same way, when we just exchange the male plant with female plant, that is dark green leafed male plant and pale green leafed female plant, when they both are crossed, the offsprings or the individual produced in the F1 will be a pale green leafed plant, which is a characteristic feature of the female plant. So irrespective of the color of the male plant, what is present in the female plant alone will be inherited in the progenies for the color of the leaf, because these genes are present in the chloroplast, and this is the inheritance of leaf color in Mirabilis jalapa. Now let us learn the mitochondrial inheritance in pearl maize. There are two types of pearl maize. What is pearl maize, which is called as kambu in Tamil? So two types of pearl maize plant with normal cytoplasm, which is considered as fertile, and pearl maize with mutant cytoplasm, yes, which is considered as the sterile. And this is the best example for cytoplasmic male sterility inheritance. Cytoplasmic genetic male sterility. So this is actually recently discovered. Cytoplasmic genetic male sterility is common in many plant species. And there are two types of cytoplasm, normal cytoplasm and sterile cytoplasm. Normal is denoted by N and sterile is denoted by S. And the gene which is responsible for restoring the fertility is considered as capital RF. Now we are going to cross two parents, a male and a female plant. A male plant is taken with the fertile cytoplasm N, and the female parent is taken with the sterile cytoplasm. But the male does not possess the fertility factor in the fertility restoring factor in the nucleus, whereas the female possesses. So when they both are crossed, so they can produce these types of gametes. The female can produce two types of eggs with the restoring factor, without the restoring factor, but the cytoplasm is sterile, whereas the male can contribute only the nucleus without any restoring factor. So when this egg is fertilized by this male gamete, the individual produced will be a fertile male because of the presence of the restoring factor. Whereas when this egg is fertilized by this gamete, male gamete, the individual produced would be a sterile male due to the absence of the restoration factor. So the restoration factor capital R determines the fertility of the individual produced. And moreover, the male gamete contributes only the nucleus and the female gamete contributes the nucleus and also almost all the cytoplasm. What is atavism? Atavism refers to the reappearance of the ancestral characters. So what our forefathers have got, those characters, if they were absent in our present generation, they may be reappear in the future generation. And this is what is referred as atavism. And so atavism is nothing but refers to the reappearance of the ancestral trait that has been lost during evolutionary changes in the previous generation. The best example for this is re-emergence of the sexual reproduction in the flowering plant Ieracium pilosella.